So we've got two really exciting talks today. Our first speaker is Dr. Nerea Irigoyen. She did her PhD in the Spanish National Biotechnology Center under the supervision of Professor Jose Francisco Rodriguez Aguirre and Professor Jose Ruiz Caston. And then she moved to the University of Cambridge, where she's done a postdoc under the supervision of Professor Ian Brierley. Currently, she's a research fellow or junior PI at the Division of Virology at the University of Cambridge. So over to you, Nerea.
Okay, thank you very much, Erica, for this nice presentation. See if, okay. You can see it, right?
Yep, that looks good.
Okay, perfect. So I'm just going to put the laser pointer. Yeah. Okay, so thank you very much for the introduction, Erica, and good afternoon to everyone. Good morning for people in a different zone. I'm going to be working about work that we started some time ago on the manipulation of the unfolded protein response and how we can use this as a pharmacological strategy against coronavirus infection. So actually, this started very, very long time ago when we started to apply this technique, which is called ribosome profiling, that can be described as the global analysis of translation. So we were the first one to apply this technique to cells infected with an RNA virus, and that happened to be the coronavirus mutant hepatitis virus MHV. So there was published back in PLOS some years ago, and I need to say that I've been very happy during the pandemic because this paper has been highly cited and has become a kind of a seminal paper for checking translation and transcription of coronaviruses such as SARS-CoV-2. So another thing that I don't probably need to talk about much in this forum is that we use MHV, that actually is a beta coronavirus that happens to be in the same genus such as some other very important human pathogenic coronavirus such as SARS-CoV-1, MERS, and SARS-CoV-2. So what is ribosome profiling? So the technique can determine the position of a translating ribosome through a discrete 30 nucleotide fragment that is protected from nuclease digestion. So how do we do this? So we have our cells that are going to be infected, and then we will freeze the cells, and we will have two different sets. In the first one, we will extract total RNA, and that will be subjected to RNA sequencing, and that will provide information about the transcriptome. And then in the second set, we are going to extract these polysomes that will break up into ribosomes by the addition of an RNase one that can basically chew the mRNA in between ribosomes. And then we will isolate these monosomes, and what we are going to be really very interested is in this mRNA that is actually being translated. So this is what we call ribosome protected fragment, and it's normally 30 nucleotides in length. Then we will add the different adapters to have our library prepared for Illumina sequencing, and this is how we obtain what we call the ribosec libraries that will provide information about the transcriptome. So which are the applications of this technique? So first, we can check for the translational activity of the transcriptome. We can also identify novel short open reading frames. And this is something that we have just done with Zika virus. And if you are interested, we have just posted this paper in my archive. Also, if we are using different drugs that can hold a translation initiation, we can check for initiation sites, even non-canonical ones. And if we have very good phasing in our sample, we can even identify overlapping open reading frames, saying that we can distinguish which the ribosome, which frame is actually translated. And also when we are combining the analysis of ribosec and RNA-seq libraries, we can check for the analysis of global gene expression. And this is something that we've done with coronavirus. So this part of the work I'm presenting has already been published. That was in PLOS last year. And that was when we were analyzing the global cellular gene expression using the combination of RNA-seq and ribosome profiling when we were comparing mock-infected cells versus infected cells. This work was led by two people, the former postdoc Liliana Chavarria and also a former PhD student, Georgia Cook. So when we wanted to know which were the cellular pathways that had some biological relevance during infection, and for that we performed what is called the reactant pathway enrichment analysis. And then we check that most of the upregulated pathways here in orange were related to the unfolded protein response. Also, ATF6, as I will say later, is a branch or a pathway of this UPR. And also the most downregulated pathways were actually related to protein synthesis. Also taking advantage that we can also check, because this is RNA transcription, so we wanted to check what was really happening at the level of translation. In this plot, what we are seeing is what is called the translation efficiency, which is just basically the number of RPFs, it's just basically translation divided by the transcription. So in this volcano plot, we can see that most of the upregulated genes at the level of translation were also related to the unfolded protein response, such as ATF4, ATF5, GAT34, and CHOP. So you all know that the endoplasmic reticulum is the major site for synthesis and folding of secreted and transmembrane proteins. However, when the protein synthesis surpasses the folding capacity, all these unfolding proteins will accumulate and they can activate what is called the ER stress. So in these situations, there are going to be three sensors, PERK, ATF6, and EDA1, that will transmit the signal to the cytosol and the nucleus, and then the cell will try to lower the protein synthesis and increase the ER folding capacity. And in this situation is when we can say that the unfolded protein response, or the UPR, has been activated. Coronaviruses, during their life cycle, they can activate the UPR at different stages. So the first one is during virus replication, and more specifically during the formation of the double membrane vesicles, which are the place of the viral factories. And this is because the virus, in order to form these TMVs, are going to be using membranes that are deriving from the ER. So the UPR can also be activated during infection, during the massive production and modification of the structural proteins. And it can also get activated during virion assembly, during the depletion of the ER membrane during the budding before exiting the cell. So as I said, MHP can activate the UPR, and the UPR consists of three different branches. So at this stage, we really wanted to know which were the branches of the UPR that were activated and to what extent. So first, we monitor the first branch, which is going to be the EDA1 exit branch. And in this situation, upon ER stress, EDA1 will oligomerize and will become activated. And the RNA activity of this protein will be able to remove a 20-nucleotide intron, making this and splice XBP1 in what is called the splice XBP1, that when translated is a transcription factor that translocates to the nucleus and can upregulate some genes related to ER chaperones and ER components. So to monitor this during infection, what we did was to infect our cells with MHV during a time course, and then we extracted total RNA, and we performed an RT-PCR, and we were checking for the ratio between the spliced version of XBP1 here, as you can see here in mock infected cells, compared to the spliced version that we see is going to be the most prominent one in infected cells from 5 hours post-infection onwards. Here we are also using tunicamycin as a positive control. So this is a drug that can activate the three branches of the UPR. And RPL19 here is used as a loading control, as this gene doesn't change during ER stress. So the second branch that we were interested in is what is called the ATF6 branch. So here, upon ER stress, ATFs can translocate from the ER to the Golgi, where it's going to be cleaved by two serine proteases, S1P and S2P, and then this N-terminus acts as a transcription factor that will upregulate the transcription of some genes, such as PIP, calreticulin, and ERP94. So we monitor this in infected cells by immunofluorescence. Here we have mock infected cells, and here coronavirus infected cells. And you can see, like, during infection, there is a translocation of ATF6 from the cytoplasm cytosol into the nucleus, as you can see here. And the last branch is the PERK EAF2 alpha ATF4 branch. And here, during ER stress, PERK will oligomerize and autophosphorylate, and this activated PERK protein can phosphorylate the alpha subunit of the initiation factor 2. And this will prevent the recycling of the EIF2 GDP, the inactive ternary complex, into the active ternary complex. So it will just basically lower the cellular translation. However, under these conditions, there are some genes such as ATF4 that can be translated, and ATF4 acts also as a transcription factor, upregulating some of its target genes, such as CHOP and GAT34. So we monitor this by Western blotting, and then here we have the infected cells, tunicamycin-treated cells, and mock infected cells. As you can see here by Western blot PERK, we had a very clear ban in mock infected cells. However, from 5 hours post-infection onwards, we start to see like multiple species in infected cells, and this is probably due to the multiple phosphorylated species, which is quite similar to what is happening in tunicamycin-treated cells. We can also see a bit of upregulation of ATF4 and phosphorylation of EIF2 alpha. So at this point, we knew that, yeah, so MHP can activate the 3 branches of the UPR, and there has been a lot of drugs trying to target this cellular response. So we decided to test some of these drugs, and we wanted to check whether they might have an effect on virus replication. So to block this pathway, we use this ERA inhibitor, the STF083010, that can block the RNase activity of ERA1. We also use this ATF6 inhibitor, the AEBSF, which is a serine protease inhibitor. And for the last pathway, we use 2 different drugs. One is going to be an inhibitor for PERK, DSK26-414, and then ISRIP inhibitor, which is an integrated stress response inhibitor that blocks the interaction of this EIF2B with the phosphorylated EIF2 alpha. So in this situation, translation occurs normal. So we use this different inhibitor at these given concentrations in cells that were infected with MHP, and then we add inhibitors after the absorption hour. Once that the cells were harvested, we titrated the supernatant, and we can see, like in all the cases, we can see a significant reduction. However, the titers were not extremely low. That was ranging between 2-fold and 6-fold. So then we decided to test what will be happening if, instead of just only using or just blocking 1 branch, if we were blocking 2 of them at the same time. And this is what we did in this experiment. And you can see here that we can see reductions of 4-fold when we are using the PERK inhibitor and ISRIP, that, remember, they're both of them going to be blocking the same pathway, to 100-fold reduction when we were just blocking the EDEM pathway and ATF6 pathway. Also, if you can see here under the microscope, in infected cells, we see the formation of syncytia. However, in these infected cells that has been treated with the different drugs, the syncytia formation is quite delayed. So at this point is when the SARS-CoV-2 pandemic kicked off. And the first thing that we wanted to check was whether this virus was also able to activate and induce the UPR, and to what extent. So we did that in a couple of cell lines, CALO3 and Vero cells. And here I'm just only going to show what the results that we obtained with CALO3, always quite similar in Vero cells. Here we have tunicamycin-treated cells, mock infected cells, and SARS-CoV-2 infected cells. In terms of the PERK EIF2 alpha ATF4 pathway, we can see a bit of phosphorylation of PERK, some induction of ATF4, and phosphorylation of EIF2, indicating that this pathway will be probably activated, not to a very high extent, but to some extent. Also, ATF6, we can see the cleavage, the N terminus of ATF6, indicating that, yeah, this pathway has been activated, and also the splicing of XBB1, basically suggesting that, yeah, SARS-CoV-2 can also induce the 3 branches of the UPR. Then we tested the different drugs in SARS-CoV-2 infected cells with an MOI of 1 and 5, and we could see some, again, reduction in the virus titers. And that was 100-fold in Vero cells, and that was even much more dramatic in CALO3 cells, where we could see an almost 1,000-fold reduction in terms of virus release. So in order to confirm this, we use 2 new inhibitors that were even much more specific and less toxic than the previous one. And in this case, we use KIRA8. This is a very, very specific inhibitor of the RNA, of the oligomerization, and therefore the RNase activity of IRE1, and also CAPin A7, which prevents the translocation of ATF6. So we tested, here I'm showing the blood that we got when we infected viral cells with SARS-CoV-2 MOI of 1 at 24 hours post-infection and also 48 hours post-infection. Here in this lane, I'm showing infected cells at 48 hours without any kind of treatment. Again, we see some phosphorylation of PERK, activation of EIF2 alpha, and also we see the cleavage of ATF6 and the splicing of XP1. Then when we are treating these cells with CAPin only that prevents ATF6, we see that ATF6 cleavage has disappeared, and something similar happens when we treat the cells with KIRA8, where we don't see the splicing of XBP1 any longer. We also treated these cells with the combinations of both of these drugs and also with the previous combination as a control. In terms of virus reduction, as we can see here in green, there is no are the cells that didn't have any treatment. Then we have the individual drugs. And here in yellow is KIRA, and also the combination of the 2 drugs here in orange, and in brown is the previous combination. Good thing is like this was maintaining time, and after 48 hours post-infection, it was a 500-fold reduction in terms of virus release, and it was much better than with the previous combination. And please pay attention that with KIRA8, we see a further reduction in virus release compared to CAPin A7. So as you all know, the virus has evolved, the pandemic has evolved, and now we have different areas of concerns. So what I'm going to be presenting now is a paper that has just been submitted and is about how the one specific arm, the IRE1 XBP1 of the UPR, is a critical host factor involved in SARS-CoV-2 infection. So what, as I have previously said, that the 3 branches could be activated, but it seems that some of them, especially the PERK pathway, is much more dependent on the cell type that we are infecting. However, this pathway, the IRE1 XBP1, is always activated, and it seems that is one of probably the one that gets activated first. We have also in our previous work, we said that when we were transfecting a plasmid encoding for the S protein, we were able to activate the 3 branches of the UPR. So the first thing that we did was to transfect a plasmid encoding for the S protein from the different variants of concern into 93 cells, and then we harvest the cells at 36 hours post-transfection. As we can see here in this plot, we see the expression of the S protein. We see that in transfected cells with the S protein, we see an upregulation of HER1 protein, which is a general marker for ER stress. And because we are interested in the XBP1 pathway, we can also see the expression, the upregulation of the expression of XBP1. We also wanted to check what is happening in terms of splicing. And as you can see here, we are using the D614G as a kind of control for the regional virus. We see splicing in all the different variants. Seems that with Omicron, BA1 and BA2, the splicing of XBP1 is even a bit higher compared to the other ones. But as you all know, not always transfecting proteins can somehow mimic what is really happening with the virus. So what we did next was to infect A549 cells with different variants of concern. In this case, was an MOI of 0.1, 16 hours post-infection, and we treated the cells also with KIRA8 to check whether we were actually able to reduce virus replication. So as we cannot see here in this plot, we have our wild type virus, and then different variants of concern in which we have untreated and treated, and in some cases we can start to see like even reduction of the S protein, indicating that somehow probably the virus replication has been impaired. And then I want you to check in what is really happening in terms of splicing of XBP1. we can see that there is a splicing with the different variants of concerns, although the splicing with alpha and delta seems to be slightly higher compared to Omicron and the wild type. So in terms of virus release, so we did the titrations of the different supernatants, and we can see that there is a reduction in the virus when we are treating the cells with Kira-8, and it's the reduction with the delta variant of concern is very, very high. And this is a bit surprising, because if you see here in terms of how XBP1 has been spliced, it seems to be quite similar between alpha and delta. However, in alpha, there doesn't seem to be any change. So that would be extremely interesting to keep studying what is really happening and which are extra factors that might be involved in alpha replication compared to delta. And these titers were somehow confirmed when we were checking for the amount of viral RNA and intracellular viral RNA. Again, we see a significant reduction in all the cases apart from alpha, where this reduction was not achieved. So just to summarize, what I have just been presenting is that the cellular response to beta coronavirus infection highlights the UPR activation as a highly prominent pathway. With that first paper, we demonstrated for the first time that SARS-CoV-2 could trigger the UPR through all the three controlling sensor pathways, and also that efficient beta coronavirus replication requires the UPR, and that could be a very good pharmacological, and the pharmacological inhibition significantly inhibits replication, meaning that this could be a very good target. And also the dual combination treatments targeting multiple branches were able to reduce SARS-CoV-2 replication between 500 to 1,000-fold in different infected cells. And also we demonstrated that mostly all of the variants of concerns can activate the UPR. So which are the implications of this? So there are some clinical manifestations of COVID-19 that do play with an exacerbated UPR response. And some of them are the endothelial dysfunction that has been associated with acute respiratory distress syndrome, thromboembolism, cerebral and cardiovascular disease, and some neurological complications, and also the pulmonary fibrosis that is produced with SARS-CoV-2. The UPR might be exacerbated as well. So we believe that the use of this type of inhibitors could have a dual therapeutic effect. Will not only contribute to the reduction of viral burden in patients, but also that can diminish the pathophysiology associated with COVID-19. So we think that targeting a cellular response, well, also know that targeting a cellular response will reduce the chances of virus escape mutants. So probably if we are using different antivirals, one is going to be targeting the virus, another one that's going to be targeting the host, that would be the way forward to treat this type of infections. And also I would like just to finish saying that the activation of the UPR is not exclusive for coronaviruses, and there are a lot of viruses, if not almost all of them, that are activating this cellular pathway. And we have very good data in which we'll probably publish very soon that Zika virus and also influenza A virus are activating the UPR, and they do respond quite well to these inhibitors. So with that, I hope that the next time that we are going to have a viral pandemic, we will be able to fill some of these shelves with more broad and spectrum antivirals that we can be using before we can have a proper vaccine. And I would like to finish this acknowledging and thanking everyone that has been involved in this work during the years, all the people at the University of Cambridge, also in Zurich, Burbright with Eric as well, University of Bristol and University of Valladolid, especially Jose Fernandez, that was doing all the variants of concern work. And yeah, thank you very much. And I will be very happy to take any of your questions.